This Week in Virology, the podcast about viruses, the kind that make you sick.
From Microbe TV, this is TWiV, This Week in Virology, episode 884, recorded on April 1st, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from New York, Amy Rosenfeld.
Hello, Vincent. How are you today?
It's April 1st.
So it's April Fool's Day.
Where
Maybe
did March
that's why
go?
we have nobody else with us.
That's it. Nobody else could make it today. It's unfortunate, but it's fine. We are in good hands here. And,
hopefully.
Maybe sometime soon we won't be saying from New York anymore, right?
Where am I going?
Oh, my gosh. We could say from New York and fool everyone. By the way, we talked about bagels the last Friday you were on.
Yeah.
And you said that the water makes all the difference. And someone sent us a long email saying why that was wrong. And I sent it to Amy and she said, seriously? So if you need a translation, when Amy says seriously or shocking, it means she doesn't believe it. Okay.
It means, why are you wasting my time with this crap? But wait, you should show the picture of all the cream cheese spreads, because that's just atrocious. The fact that there's a honey bacon sriracha spread. Where's the picture?
I could get it, but I can.
You got to show the picture. I mean, the honey sriracha bacon. I mean, if that's not trife, I don't know what is.
So this has to do with the new bagel store underneath on the ground floor, just underneath the incubator, which I was very excited to see. Last week? Last week when it opened. And I went in today to get some bagels. I got myself a sandwich, and let me show you. I took a picture of the
condiment counter.
What do you call that case there? That thing that's in there? The condiment counter, or the case, the deli case, or something, where they put stuff. Where is it?
Just call it the condiment counter.
Condiment counter.
But there should never, ever be anything that says smoked bacon and sriracha, or honey bacon and sriracha.
Let me.
That's just not good.
Let me put the camera 3 on so you can see it.
Blow it up to the one right in front that says the bacon.
So look, these are all spreads, cream cheese spreads. And this one here, honey bacon sriracha cream cheese. And Amy freaked out when I sent her this picture. Is that bad?
Well, you only eat pork when you cross the Chinese restaurant door frame. That's the only time it doesn't count.
But look at some of these others: garlic and herb cream cheese. I don't know what this one's cut off. Sun-dried tomato. That might not be bad, right?
It might be fine.
The jalapeno cilantro. Nah, that doesn't look very good. Honey walnut. Nah. Strawberry. Nah.
No, but the Nutella.
Where's the Nutella? Here, Nutella chip. Nah, that's ridiculous. Chocolate, this and that. We're getting into sweet. Walnut raisin, cucumber dill, guacamole. Here's the one here in the back, the plain. Doesn't even have a sign. Right? Because they go through it too quickly. Oh, my God. Anyway.
Well, I think we can recognize that. But the others, you can't really recognize. But
no.
I would have thought that the sriracha would have actually curdled the cream cheese. I mean, hot sauce.
Yeah, because the pH is off. Yeah.
Yeah. I would have just thought that hot sauce and cream, any milk product would have curdled. That's what I told Edmund today. I was like, Edmund, look. And I was like, I thought
you would like it,
I do. I think it's hysterical.
Anyway, the bagels are good. You have to try one. You're coming Sunday to help do some work at the incubator. Get yourself a bagel, okay, and try it. And then don't yell at me if it's bad, because I like it. If you don't like it, that's fine.
I'm not going to yell at you.
Okay.
We'll just know. What did Kathy say today when we passed her on the street? The non-Jewish bagel.
Well, it's funny. There's a sign on the wall in this bagel joint which says it has a picture of a bagel with cream cheese and lox. And it says, it's actually in French, but it says, this is not a bagel. Ce n'est pas un bagel. I don't know why they have it up there, because then they have all these spreads that are not traditional.
It's the irony.
It's the irony. The irony. Anyway.
That's why it's funny.
I'm not going to mention the name of the place, but if you know where the incubator is, you could figure it out, because it's right beneath us. It's there all the time. It's open 7 a.m. to 3 p.m. and I could go for lunch, but I usually don't eat a sandwich every day. I have yogurt, but today I ran out of yogurt, so I got a sandwich. It was so big, I couldn't eat the whole thing, so I have it for Monday. All right, we're actually going to do some science today.
Oh, really? Why start now? 10 minutes into the podcast.
First paper is
start now?
From Cell Reports. Asthmatic bronchial smooth muscle increases rhinovirus replication within the bronchial epithelium. This is from a group in a variety of places in France. Pauline Esteve is the first author, and then Thomas Trion and Patrick Berger, two co-corresponding authors. And I thought this would be interesting because Amy works on rhinoviruses, and rhinoviruses are major contributors to induction of asthma, right? So asthma, of course, is an airway disease. Very common. 6 to 20% of Western European countries have asthma, and viral infection of the epithelium in the bronchi has been implicated in 80% of asthma exacerbations in children and adolescents and 60% in adults. And in each case, rhinovirus was the dominant viral pathogen, causing 60% of all virus-induced asthma exacerbations. What is it? And so you are interested in asthma because of the rhino connection, correct?
There's also an EBD68 connection.
So enterovirus D68 may also exacerbate asthma?
Yeah.
Okay. Well, this paper is of interest to you since you work on both viruses, right?
Yeah, I'm very interested.
They also say that the susceptibility of bronchial epithelium to rhino infection appears to be increased in people with asthma, but the mechanism is unknown. And that's what this paper explores. And they explore something called bronchial smooth muscle. And in people with asthma, the mass of this bronchial smooth muscle appears to increase, and it signals a poor prognosis because increased bronchial smooth muscle is associated with decreased lung function. So, Amy, what is bronchial smooth muscle? Is that beneath the epithelium?
Yeah, it's underneath the lamina propria. It's the next layer underneath the lamina propria. But it's also really interesting because, like Palmenberg and Rhino C, they feel that the connection to asthma exacerbations are all about a point mutation in the receptor, which sits on the epithelium. Which
causes higher levels of the receptor on the plasma membrane, right? And therefore more infection. Mm-hmm. Now, the muscle, this bronchial smooth muscle, is this involved in lung contraction and so forth and breathing?
Yeah, but the bronchia aren't lungs. So like your trachea bifurcates into two arms, right? Yeah. Two bronchi. And then they split off and get divided and subdivided and stuff. And these are like the channels. And then as you become stressed or infected, the muscle contracts them and it makes it difficult to breathe. And so,
so that's why expanded.
That's what asthma really is a constriction of the
bronchi. So that's why increased mass is bad, because it makes the opening smaller. Mm-hmm. What's the normal function of this muscle underneath the bronchial epithelium?
Well, it regulates like how much air you take into, probably. You don't. It talks about lung volume capacity and tidal breath capacity. And so this regulates all of that. Because sometimes if you have too high an oxygen content, I think it's acidic. Your blood becomes 2 pH. And if you have too basic, and if you have too much carbon dioxide, it becomes acidic. So it helps regulate, your respiratory rate.
All right. So in this paper, they want to know if there's some role, if there's somehow rhinovirus and the bronchial, the BSM is involved. And so they use a very interesting cell culture model. It's a
really
co-culture
cool.
where they have, so these are called air-liquid interface cultures, where you have a cell culture dish with a membrane across it suspended on top of the plastic bottom. So you could have, normally what you do is you would explant respiratory cells onto the membrane, and they would be covered with medium. And then when they're confluent, you take the medium off, and then they would differentiate into airway epithelium.
The media is on the bottom.
Right. And with the medium on the bottom and the air on the top, it becomes like your lung, right? Where the epithelium is exposed to the air.
It becomes your bronchial epithelial lining,
Right.
not your lung. It's different.
Sorry, I keep saying lung because I view it as all one thing. But yes, I know.
Yeah, but it's not. It's not all one
thing.
thing.
You could just keep correcting me. Now, in this case, underneath, they put BSM, okay? They put BSM cells, bronchial smooth muscle. I'm going to call it BSM from now on.
Yeah.
From non-asthmatic or asthmatic patients. And on top, the bronchial epithelium is just from non-asthmatic patients.
For the majority of stuff, but they do use epithelial cells from asthmatics.
Yeah, it's kind of a control at some point or an experiment to figure out.
Right. So they first do it with the non, and then they test to see if the asthmatic not an epithelium makes a difference, and they find that it doesn't. So it's easier to do it with the non-asthmatic.
So they have on top of the membrane the epithelium and underneath the muscle cells. And then they can infect the bronchial epithelium with rhinovirus, and they use a multiplicity of infection of 0.1, which they say emulates in vivo infection. So low multiplicity. You're not going to have a lot of viruses per cell.
Yeah, you're not going to get a multiplicity of 20 or 100.
No, it's silly to use that if you're trying to emulate what happens in the lung, wouldn't you say?
Yeah, they point that out.
All right, and so then they measure rhinovirus production.
First they measure entry, right?
That's right. They measure entry
because they want to know if being asthmatic alters the efficiency of entry. So, ICAM isn't always on the surface of the cells. Its production is stimulated by a stressor, right? Yeah. So one might think that being asthmatic, you'd have a lot more ICAM, and therefore you'd facilitate entry, right? And they first show that there's not a change in ICAM, and there's not a So asthma, their predisposition to rhino infections isn't because they're more infectable.
Yeah, they do a short infection where the virus would just bind and get in, and then just measure it to get that answer. Yeah, that's absolutely right. And then they let the infection go longer, 24 hours, and they compare the number of virus particles with either asthmatic or non-asthmatic BSM cells underneath. Mm-hmm. And you get more virus when you have asthmatic BSM cells under the epithelium. Right. It was a pretty straightforward result. And if you put non-asthmatic BSM underneath, you don't get an increase in infection. It's quite clear. Right. Mm-hmm. All right. So then the rest of the paper is trying to figure out what's going on here. It's an interesting observation. So they measure IL-6 production, interleukin-6, by the epithelial cells co-cultured with the asthmatic cells and in response to virus infection. And the IL-6 levels go up, but the IL-6 goes up even more in the supernatant of infected epithelium when you have asthmatic BSM underneath. So IL-6 is a pro-inflammatory cytokine, which is made in response to infection, correct?
Yes. It's well known that it's made in response to infection. It's been observed previously.
Yeah. And importantly, they look for cell death, apoptosis, and they do not see an increase in apoptosis, which might explain the increase in rhinovirus replication. So cell death does not account for that.
Yeah. So apoptosis was only observed when you used like an MOI of 20, which is totally artificial. And it's unfortunate that that group published that result, because it's well known, if you look at some other data from other labs, that there's no damage to the epithelial lining. And very rarely, is there cell death. And the virus, in some cases, the virus doesn't actually explode the cell. It just kind of like buds off, if I remember correctly.
It's interesting. So
so like when you look at like work by Gern and Palmenberg and Uri, they do the airway liquid cultures for their rhinovirus, and they just see the cell like pop out, and then immediately the monolayer is repaired. Like you mean
the cell is shed from the monolayer?
Yeah, it just pops off.
The virus-infected cell pops off, right?
Yeah, just pops off.
It doesn't break. It doesn't break.
Sometimes it doesn't break. It just pops off, according to them. And then the neighboring cells like move over really rapidly to fill in that gap.
Interesting. What happens if this happens in a person? What would happen to the popped-off cell?
You cough it up.
That's the end of that.
Swallow it, right?
So, and that's probably not going to cause asthma per se, right?
It depends.
Coughing up, popping off of cells?
Yeah, because, it stimulates mucus production and stuff. So it depends.
So what's the mechanism? If it's not, what is the mechanism? So they look at interferon response, and they didn't see any changes in interferon beta production or some interferon-stimulated genes. As you may remember, an early response to virus infection is the production of interferon, which in turn turns on a number of genes in the cell whose gene products have antiviral activity. Those are called interferon-stimulated genes.
Yeah, but my problem is that they looked at beta. This is a mucosal surface. You should have looked at lambda.
Yes, agreed. And they only looked at 3 interferon-stimulated genes, right?
Yeah, well, these are the 3 most popular ones, apparently.
So then they turned to another interferon-stimulated gene, PKR, which is funny. They didn't look at it initially, right? Because if they're so interested in PCR, they should have. PKR. Anyway, this is a interferon-induced double-stranded RNA-dependent protein kinase. Really important antiviral protein. Essentially, when double-stranded RNA is made during virus infection, which happens often in various virus-infected cells, PKR is activated, and it phosphorylates EIF2 Alpha, which is a translation initiation protein, and then it shuts down protein synthesis.
Yep.
And it's a way of turning down virus synthesis in the infected cell. Now, what they find is that the production of PKR was decreased in bronchial epithelium when you co-culture it with asthmatic BSM cells and then infect with rhinovirus, compared with the non-asthmatic BSM. All right, so PKR is decreased, and they went into people and they got bronchial epithelial brushings, and they do proteomics. Did they do mass spec here? I presume so, mass spec.
Yeah, I would assume.
And they've showed that even in people with asthmatic, so they have these brushings from asthmatic and non-asthmatic people, and PKR decreases in asthmatic bronchial epithelium. So this is a real thing that happens. So it's PKR that seems to be maybe, well, we'll see. We'll see if it is actually PKR. Let's see. What's this double-stranded RNA here? What is this? So they then say, if PKR is decreased, then we should see a decrease in the phosphorylation of EIF2 alpha. This is an old favorite of ours, Amy. We used to work on EIF2 alpha years ago.
I used to work on translation years ago.
Well, yes, you, and I have to be careful what I say because you're listening like a hawk.
I was going to say, and yes, they do mass spec proteomic analysis. Yes. And my friend still works on PKR and EIF2 alpha phosphorylation. So.
But Mauro?
Yeah. He still is heavily into EIF2 alpha phosphorylation.
Yeah. Mauro Castellmaterli, who was on TWiV, right?
Yeah.
Okay.
But he discussed something separate when he was on TWiV. He discussed his microbiome stuff.
Microbiome and?
Autism.
Autism, right. So they find the level of EIF2 alpha phosphorylation is decreased in these virus-infected bronchial epithelium co-cultured with asthmatic BSM cells, and the rate of protein synthesis, it's slightly decreased, which is not really consistent, because if you decrease phosphorylation of EIF2, you should get more protein synthesis, right?
Yeah.
Because phosphorylation is what...
Wait a minute.
You say, we observed a slight but significant decrease in protein translation.
You can't, so right there, the terminology is wrong. You can't translate a protein. You can only translate mRNA.
Yeah, I know. I noticed that too, yeah.
I was like, maybe it's a French thing, but you can't translate a protein.
What does it mean, a slight but significant decrease? What does that mean?
I don't know.
They don't apply any statistics here, do they? Let's see, that's figure...
3E.
3E, where is that?
Yeah, they apply some statistics.
They have error bars, and then they
the standard error of the mean
p-value. Well, I don't think that means anything, but I think the key here is that EIF2 alpha phosphorylation decreases, and that's fine. All right,
yeah, but there is still a slight band. You can see it on the gel.
Yeah. All right. So what's the mechanism? Okay, so in the old days, we would plan some experiment, but now what you do is transcriptome and proteome analysis. You look at everything.
Well, wait a minute. Didn't they treat...
unbiased transcriptomic and proteomic analysis.
Yeah, I just got confused which order they did it.
From these BSM cells. And they see differences, and they chose a molecule called CCL20, which its mRNA appears to be highly overexpressed in asthmatic BSM cells compared with non-asthmatic BSM cells. So CCL20 is macrophage inflammatory protein 3 alpha, MIP3 alpha. And so it is up in the asthmatic BSM cells. And this is a secreted protein, right? So the idea is that, if it's secreted, it could make its way into the epithelium, right?
Yep.
And it's nearly absent in the supernatant from non-asthmatic BSM or BSM cultured alone. That's interesting. I guess without rhinovirus infection, right?
Right.
So somehow rhinovirus infection is leading to an increase in the production of CCL20 from these BSMs. And they do look in lysates from asthmatic and non-asthmatic persons, BSM lysates. How would they get BSM from people?
You scrape.
A scrape, yeah. Mm-hmm. So they make lysates and they do ELISA, and they see increased CCL20 in these lysates from asthmatic BSM. It's nice that they correlated cell culture with people data, right?
I think it's important. Yeah. Because there's no animal model for rhino, so it would be hard to validate this. Yeah. You need to show that it actually occurs in some host. Yeah.
So CCL is up, PKR is down, so maybe there's a connection. So now they're going to explore that. Mm-hmm. So next set of experiments, they make recombinant CCL20, and then they add it to bronchial epithelial cells. Now, there are no BSM cells anymore. They just add the protein to the epithelial cells, and then they find that the PKR, mRNA, and protein levels decrease in the presence of rhinovirus infection.
Yeah.
And it also, adding CCL also inhibits phosphorylation of PKR, which is a way it's activated to be able to phosphorylate EIF2 alpha.
Yep.
Isn't that interesting? And also EIF2 alpha phosphorylation was inhibited following addition of CCL20. So it's a very nice experiment. I guess there's a receptor on the cell for CCL20, right?
Yes.
And it must be signaling in such a way to decrease the production of PKR, correct?
Yes.
All right. So then they did an experiment with antibody to CCL20. So you have bronchial epithelium that you infect with rhinovirus, and then you add CCL20 antibody in the supernatant. This is really interesting. The addition of antibody to CCL20 blocked the increase in rhinovirus production in co-cultures with asthmatic BSM cells.
Which is what you expect.
Right.
Yeah.
However, they were unable to show that blocking CCL20 rescued PKR activity. It's interesting, right?
Yeah.
So maybe the PKR is not relevant to CCL20, even though it makes a difference. Maybe I don't know. That's a puzzle there. And the reason for that is because CCL20 increased the number of particles in the bronchial epithelium.
Yep.
All right. So then how does CCL20 work? There is a downstream kinase. There's several downstream kinases that they looked at. MAP kinase, PI3 kinase, J and K kinase. They have inhibitors of these. 3 different inhibitors. And so MAP3, MAPK and PI3K kinase inhibitors had no effect, but the inhibitor of J and K, the J and K Janus kinase pathway, abolished the effect of CCL20 on PKR expression in the bronchial epithelium. So the J and K pathway is a signaling pathway involved in inflammation.
Yep.
And then they cut out the J and K gene with CRISPR. And so you get less J and K in the bronchial epithelium. And now when you add CCL20, there's no effect on PKR expression.
Nope.
So basically, when you add CCL20, you're going through the J and K pathway to reach PKR, right?
Yes. It's not direct, yeah.
And then they cut out the PKR gene with CRISPR, and you can also get an increase in rhinovirus particles, which is not surprising, right? Because PKR is antiviral.
Right.
Correction. So that's the paper. Those are the data. You have CCL20 is produced by the BSM. It gets into or binds to receptors on the bronchial epithelium, and then decreases PKR and increases rhinovirus yields.
Yep.
And so, Amy, why do you think that asthmatic BSM makes much more CCL20?
It's stressed. Asthma is an inflammatory disease. So.
All right, so that's part of the stress response. They're making a lot of CCL20, and that inhibits PKR.
Yeah.
Now, why would CCL inhibit PKR? What would be the physiological role of that?
Well, you want to turn off so PKR also regulates interferon gamma production.
Mm-hmm.
So you might not want to have a lot of interferon gamma production, right?
Yes. Yes.
So, one thing that they should have looked at was the levels of interferon gamma.
Yes, I agree. And lambda too, right?
Yeah.
Yeah.
And
lambda, because lambda is an epithelial barrier interferon, right?
Yes.
Mm-hmm.
It's the right interferon. And gamma is necessary to recruit T cells and stuff.
Mm-hmm.
And it's downstream of PKR. So
yes, would be interesting.
Yeah.
Yep.
So you should have looked at it, but you can't do everything in all.
Now, they say there are other data that have been published which implicate, along with their data, CCL20 as a important cytokine in asthma. And it turns out that some CCL20 or CCL20 inhibitors are in early phases of development for treating people.
Yes.
And they may be useful for asthma. That would be exciting, wouldn't it?
Yeah, it would be really exciting.
They say it's in development for cancer and other inflammatory diseases.
Yes.
But I can imagine they might have side effects, right? If you give people CCL20. So
I'm sure.
You'd have to balance that, right?
I'm sure you want to balance it. But they say it's been known to be increased in asthmatic spectrum and that
Sputum, yeah.
Sputum. Thank you.
They also say there's little evidence for a role of PKR in asthma.
Yes.
Only a single nucleotide polymorphism in the EIF2 alpha gene that's been associated with asthma has been identified. But maybe you just need one, right?
Yeah, but it's not EIF2 alpha. It's EIF2 AK2. It's different.
I'm sorry, that encodes PKR. Yeah.
EIF2 alpha is different.
I misspoke again. Yeah, PKR. But so there is a SNP in PKR gene that associates with asthma.
But asthma is very complex. So you have that, and then you have CDHR3 from Anne and various other things. And I'm sure, we'll find out that there is an MDA5 SNP, because MDA5 SNPs have been shown to moderate rhinovirus and enterovirus infection by Casanova.
Right.
Yeah. So it's not a single gene phenotype, right? Very rarely are diseases single genes. They're genetically complex. Isn't that how Barrick phrased it?
Don't remember. I'll take your word for it.
Yeah, I think that's how he phrases it when he describes using the collaborative cross mice to tease it all out. Is that they're polygenic.
Mm-hmm. They say we don't know the mechanism of decreased PKR in response to CCL20.
Right.
By the way, the receptor for CCL20 is CCR6. And I would guess that could be involved in asthma as well, right?
Yeah, I mean, so they only look at ICAM, right? They don't look to see if CCR6 is upregulated or downregulated.
That's right. So it could be.
You can imagine that it could be.
Yeah. And CCL20, if you're interested, it favors chemotaxis of immune cells to the area whatever the area is that's a problem, including a variety of T cells, B cells, dendritic cells. And this has been observed in cancer, psoriasis, and rheumatoid arthritis.
Yep.
But how this CCL20 CCR6 interaction downregulates PKR, they don't know. Although you could figure that out, right?
Yeah, for sure.
Right, if you wanted to do that. And I'm sure they are. I'm
sure that they're working on it.
So did you find this an interesting observation?
I think it's interesting for sure. I think that appreciation, so we focus heavily on the direct cells that are the sites of infection or the tissue that is the site of infection. And we don't really understand the interplay of the complexity of the organ. Nobody, you have smooth muscle totally throughout the gastrointestinal tract. Like, what's its role in regulating, neurovirus and stuff, infection or whatever, pathogenesis, or a rotavirus, right? We're very heavily focused on the immune infiltrate right into the epithelial layer. We don't really look at how, tissue homeostasis alters its function.
Yeah.
And then when we make these artificial systems, we totally just look at the layer. We just look at the epithelial layer.
I think that's a really very good point. And the reason is that we develop cell culture systems to grow our viruses and study them. And, their development in the 50s was very exciting. We became enamored with them. But then, as you say, we ignore the fact that in an organism, there's a complex tissue architecture. There are other cell types next to the ones that are being infected.
Well, it also has to do with our inability to generate the complexity of the tissue, because when you take a stem cell, you can only differentiate it into one type of germ layer, right? You can't. So I think the epithelial is all endoderm, and the smooth muscle is from mesoderm.
Right.
After gastrulation. So we haven't really figured out how to merge those two. And if you have, so let's say you can make muscles, you can make muscle-like cells from iPS cells. Smooth muscle and skeletal muscle and cardiac muscle are all different, as well as the smooth muscle in the respiratory tract is different than the smooth muscle in your intestines.
Right.
So we don't really know enough about how to program those cells and have it coordinated with the epithelial lining. Plus, we don't have a way to make the basal lamina that is separating all of that.
Correct.
So there's a lot to be done. And this is kind of why people think I'm anti-organoid and anti-3D cultures. It's because I'm sitting there saying, well, you don't really have the right thing. So we can't really say that you're using this to do that. And so I think if you state it properly, it's fine. But if you automatically assume, you leave out a few little words and stuff, you give the reader the wrong impression.
So I just want to say that, when we first developed cell culture in the 50s, one of the main cell lines developed was Hela cells, and it allowed us to study virus replication and the different steps of replication. But now we are trying to understand, as Amy has pointed out, how different tissues interact in a virus infection. And there's no more role for Hela cells in that. All right. Hela cells are not the cell that is infected by rhinovirus. It makes zero sense to use them to try and understand the pathophysiology of rhinovirus infection. It's fine if you want to figure out how the RNA replicates, but the kind of study that is in this paper, no Hela cells.
Yeah, but like here, for instance, a lot of the interferon stimulated gene stuff, the screens that Scoggins and Charlie did, they're all in 293 cells.
Yeah, it's ridiculous. I agree.
I mean, they're a good starting point, but I don't know.
They're a good starting point.
I don't know that I would have said that they show everything. And people have taken them to say, well, because we see MDA5 regulating picorna function, innate immunity against picornas and 293s, that's the way it works, and we don't need to investigate anything else. And it turns out that for many times, MDA5 was not was not the whole story.
Yes.
So for rhino, and so for recently for Casanova and the woman who studies 71 in Taiwan, I forgot her name at the moment, escapes me. Their paper doesn't talk about MDA5, talks about TLR3 and SNPs and TLR3 being important and stuff. So it's kind of not correct to say that we know everything from 293 knockout studies.
Yep. Agreed.
But reviewers,
Yes.
They assume that whatever is in nature and was in a 293 cell is tells us the whole story.
That's right.
But it doesn't. I
Nope,
mean, there's
nope.
still a lot to learn. I'm sure that this is just the tip of the iceberg because asthma is super complex. And so there's lots of other things that go into it.
Yeah.
I wouldn't be surprised to find out that some of this, if you told me CCL20 also had a role on, the sphincter muscles of all of these little bronchi and stuff and contracting them and stimulated the neurons or whatever, I wouldn't be surprised.
All right, let's go on to our second paper. This is a MBio paper. Viral mimicry of interleukin 17A by SARS-CoV-2 Orphate. This is from a group of authors from the Cleveland Clinic, from Korea, from Germany. The first author is Shin Wu, and the last author is Jae Jung. And so this is a SARS-CoV-2 paper. And perhaps some of you remember that severe COVID is associated with a cytokine storm, an overproduction of inflammatory cytokines and hyperactivation of immune cells. And inflammatory cytokines include IL-6 that we just talked about and others.
But severe COVID also had the coagulation.
Coagulation as well, yes. Acute respiratory distress, acute lung injury, and the storm is playing a role in that, and as does coagulopathy, for example. Now, interleukin 17A is a cytokine. It's secreted from cells, as many are, that they tell us is crucial for triggering cytokine storms. It's associated with alveolar inflammation, the destruction of the lung, and acute respiratory distress. And increased IL-17A has been found in patients that are infected with SARS-CoV, first SARS-CoV, MERS-CoV, and SARS-CoV-2. So the idea has arisen that perhaps, activation of IL-17 is part of this cytokine storm in severe COVID syndrome. So IL-17. There are 6 IL-17 family members. It's a happy family. And each of those binds a receptor on the cell surface, and there are a bunch of those. And the receptor is typically a dimer, 2 proteins. And you get different combination of dimers, of monomers to make receptors that bind the different cytokines.
Yeah, but I think one of the monomers is always the same.
What's that?
I thought A was always the same and the second one was different, like C, B, D.
That's correct. A is always there. The RA, IL-17 receptor A is always there, and then it dimerizes with something else to bind one of those IL-17. That's correct. And when IL-17 binds the receptor, the dimer, you then get downstream signaling and the eventual activation of transcription factors like NF-kappa B, which goes in the nucleus and then turns on the synthesis of the transcription of chemokine and cytokine genes. The idea is it's a response to infection, and those cytokines are helping to clear the infection. But sometimes you get aberrant activation through these IL-17 receptors, and you make too many cytokines. And so IL-17 receptor is a target for a number of diseases like autoimmune disease, psoriasis, for example. And there have been some tests of treating patients with antagonists of the cytokine itself. Yeah, I think they've even done some tests with COVID patients, right?
Yeah, but it was also observed that people who were being treated with IL-17 antagonists when they got infected with COVID had mild symptoms.
That's right. So these are people being treated for like psoriasis, right? Yeah.
Yeah.
And it's like an accidental finding, right? Yeah.
Yeah. Yeah.
All right. So that's IL-17. So
But what is Daniel's favorite anti-something or other, anti-pro-inflammatory? He has or is it IL-6 that he keeps talking about?
I don't know. It doesn't ring a bell.
He's talked about using it as like an antibody against it.
Oh, IL-6, yeah.
Oh, yeah. Okay. So it's IL-6.
So very early on, remember, IL-6 levels were known to be up. And so people, there's a monoclonal against IL-6. What's it called? Haven't talked about it for a while.
Yeah.
IL-6.
Like Tosi or something. Or
Tocilizumab, right?
Yeah. Something like that.
So I think there's one against the IL-6 and one against the IL-6 receptor, right? They're two different therapeutics.
Yeah. I mean, this is beyond my attention span, but yes.
Amy, nothing is beyond your attention span.
But yeah, I think he talked about a lot about, and I think he still periodically talks about using Tosi.
Yeah, Tosi is used to treat rheumatoid arthritis.
Yeah, and you had to use it late, not early.
Yeah, for COVID, right?
Yeah.
So Tosi is an anti-IL-6 monoclonal. That's right. We haven't talked about that in a long time, I suspect, because
we've got better interventions now I think we can use earlier.
Yes, that too. But part of the issue with IL-6 is that it was too specific. You needed a broader, because there are multiple cytokines involved, and IL-6 alone is not enough. And that's why steroids dexamethasone at the right time turned out to be useful, because it's more broadly acting, right? More broadly suppressive.
Yeah.
All right. So that's IL-17. It has some role in severe COVID. Then the virus side, the protein in the virus is encoded by open reading frame 8. That's why we call it Orphate. And Orphate is a very interesting protein, because in SARS-CoV outbreak back in 2003, that virus actually had an Orphate open reading frame, although Amy tells me it's a very different protein from the Orphate of SARS-CoV-2,
right?
Yeah,
it's hugely different.
And so, interestingly, about halfway through SARS-CoV outbreak, a deletion virus arose that lacked Orphate, and then that virus predominated for the rest of the outbreak.
Yeah, but the outbreak was 8,000 people.
Yes, it's a bit different, isn't it?
I was going to say, let's get real here.
Yes, that's true.
We're like, what, 100 to 1,000-fold?
Yeah, it's a lot more, for sure.
You think?
Now, there have been deletions of Orphate. We're not good at the math. there have been deletions of Orphate in SARS-CoV-2 that have been observed.
Yeah, very early on, too.
Yeah, and some of these, there were some papers. I remember a Lancet paper out of Hong Kong saying people who were infected with this Orphate deletion variant had milder disease.
Yeah, it was lympha.
Was that lympha? Yeah.
Right. Yeah, it was lympha.
But Orphate deleted SARS-CoV-2 do not predominate. Right? They don't seem to.
Well, it's not clear. So a lot of the variants have Orphate alterations. But we don't really know what they do, because we're so focused on the spike alterations.
But also, it's fair to say that not every isolate of SARS-CoV-2 lacks Orphate, right?
Well, they might not lack it, but they generally have, a lot of them have this L84S alteration.
So the L84S amino acid change, right? And it's also associated with milder disease, right?
Right. So, but we're so focused on spike and the spike alterations. That, I mean, who knows? You have to actually do the experiments. I know it's a novel concept.
You can't just talk about it?
Talk about what?
The experiment.
No, isn't there a song? Isn't there like a pop song from like the 80s about talking about it? Yeah, there's some pop song that's in the back of my mind from like, I don't know, the 80s or 90s.
I can't help you.
That's okay.
So Orphate is a secreted protein, right, Amy?
Yeah.
And
secreted.
It is an immunoglobulin-like protein, right?
Yeah.
Ig-like protein.
Yeah. It has a lot of different functions, or a lot of different, anti-immune functions or antagonistic. roles to the immune system have been attributed to it.
And it's they call it here in this paper highly evolving because you see a lot of changes in it in the variants. So in this paper,
it's also changed tremendously from Orphate of SARS-1 in 2003, which was membrane. It had a transmembrane domain, if I remember correctly.
Yeah, only 30% amino acid similarity with SARS-1 Orphate.
Yeah, I told you, totally different protein.
But 95% with RATG coronavirus Orphate.
Yeah, but we don't study that.
No, we don't study that. Oh, it would be interesting to look at Orphates from various SARS-CoV-2-like viruses, right?
Yeah, I believe that was a grant proposal that nobody was excited about but you and me.
Yeah,
and Rod.
It's true. Amy and I and Rod Sina Klumbu.
And actually got a fundable score, and then we were told no money.
Yeah, it's just a lot of BS. Okay, so we wrote a grant to work on Orphate, and it got a really good score. And the NIH said, sorry, we don't have any money for this. It's just BS. Okay,
I was so proud of that grant.
They gave lots of money to clinical trials of all sorts, but really good basic science. Yeah, very frustrating.
I know, and I was proud of that grant.
It's a good grant.
Really
you wrote it. You came up with the idea very quickly. We got Rod interested. Yeah. Well,
he was really excited. I could have turned him into a virologist.
Yes, Rod is a yeast guy. Yeah.
Yeah. He was on the verge. And then he pulled back. Not so good.
Well, it's partly our fault, but that's another story. All right. So then here are the experiments. They have, they're looking for Orphate in lung bronchial epithelial cells. No, not.
Well, it's known that there are a lot of antibodies directed against Orphate. Like Nisha and Anne have done these peptide ELISAs,
people make antibodies to Orphate, right?
Yeah.
Yeah. Okay.
And so they've been found using Nisha and Anne's peptide arrays, where they pass the sera from infected people over it.
Yeah.
Yeah.
But Amy, they say in this paper, in lung bronchial epithelial cells. You just told me the bronchial is not the lung. Why do they call them lung bronchial epithelial cells?
I don't know. But the lung is different.
So it's just bronchial epithelial cells.
Yeah, it depends on where you take it from.
Anyway, so they have these from ferrets and mice who are infected with SARS-CoV-2 or the supernatants of cells infected with virus, and they find Orphate in the supernatants. But of course, if you infect with a variant lacking Orphate, you don't find it. That's obvious.
Right.
So the proteins were created. They have serum sampled from hospitalized COVID patients, and they measure Orphate in the serum. And in the hospitalized patients, it's about 1 nanogram per milliliter. But in ICU admitted patients, it's 400 nanograms per milliliter. Amy, I don't need no stinking statistics to tell me that's different.
Really? We're good at this kind of math.
This is the kind of experiment I like, where you don't need to do a p-value. And maybe that's dating me. But for God's sakes, design your experiments properly. This is really good. Well, that's not fair because
they're not designed properly. The difference just is subtle.
Oh, humor me.
Okay, fine. I'll humor you.
This is not a design. They just said we're going to measure it, and it happened to be that the ICU patients, it's out of the park, right?
Yeah.
So it's higher in ICU admitted patients. Okay. So they take, they want to know if Orphate binds cells, immune cells in the blood, for example. So they take whole human blood from healthy donors and they incubate it with Orphate that they've produced in cells. And they looked at a variety of cells by flow cytometry, and they find that monocytes bind Orphate. They're the primary target of Orphate binding. Okay?
Yep.
Out of T cells, B cells, NK cells, and neutrophils, monocytes are the primary binders. All right. That's cool. So now you have a cell target.
Mm-hmm.
Then they say, what is it binding to? And other people have studied this as well. But their purified protein interacts with IL-17RA. That's the receptor A that we've just talked about for IL-17. And if you do this, so they use a cell line called THP1, which is a monocyte-like cell line, right, Amy?
Yeah,
I
but
think
you have
that's correct.
you have to be careful because it's really interesting because human cells are different from mouse cells.
Mouse cells don't respond to human Orphate, right?
Well, there's no human Orphate. There's SARS-CoV-2 Orphate. There's no human Orphate.
Yeah, mouse cells don't respond to
right, because
Orphate.
they don't produce the IL-17 receptor.
Hey, it is a human virus. I can call it. I know
it's a mouse virus. The variants infect wild-type mice.
Oh, my gosh. I can't get anything past Amy.
No, we're very, you got to be right here.
Okay. So that's correct. The mouse cells don't respond. But human cells, these monocyte-like cells, they will bind Orphate. They bind it via IL-17RA. And then when it binds, it induces phosphorylation of NF-kappa B, which is a normal downstream event from the IL-17 receptor, which we talked about just previously. And when that pathway is turned on, that NF-kappa B pathway, you get synthesis of pro-inflammatory cytokines. And there are a whole bunch of those, including IL-6. And that's why we said at the beginning, IL-17 is a inducer of pro-inflammatory cytokines. It binds the receptor and turns it on.
Well, that's why I thought like Tosi and Daniel were like connected to this.
Yeah, that's right. But here you add Orphate to these cells and you induce similar genes that you would induce with IL-17, basically.
It's mimicking.
It's mimicking. Orphate is binding the IL-17 receptor. It's very cool.
Yeah.
It's very cool. And then they do the experiment Amy just mentioned. If you add Orphate to mouse cells, it doesn't do anything. It doesn't bind it. It doesn't turn on the synthesis of these proteins. So is that possibly, Amy, why mice don't get severe COVID? In part?
Yes.
And I would say in part because I know that you don't like one answer for everything.
Well, I mean, I don't know what happens in wild-type mice. I only know what happens in some lab strains, right?
Yes.
And now, doesn't Ralph and various other people have an ARDS model?
What does it constitute?
I think that they're trying to generate an ARDS model in mice of SARS-CoV-2 infection.
Of collaborative cross mice?
Maybe.
I don't know. I know they're trying it, but I know that their passaged SARS-CoV-2, causes a more severe disease.
Yeah.
But it's not ARDS. I don't think it's ARDS. I don't think it's ALI.
What is ALI?
Acute lung injury.
Acute lung injury. Yeah, I don't know what it is exactly, but they're trying to get it to go that way, right?
Yeah.
So.
Well, maybe you should make mice transgenic for the human IL-17 receptor, and maybe they would develop some more severe COVID, right?
Well, yeah, I think if you produce the human 17 receptor on these raw cells, you do get them to turn on things.
Did they do that experiment? I don't think they did, right?
No, I don't think that they did.
But that would be a good experiment, yeah.
But you should.
Okay.
They should.
Okay, yeah, that's a good idea. All right, so then they take monocytes from people. Okay, so we've been working with a monocyte cell line. So they take monocytes from blood of people, and they add either Orphate or IL-17, and they, again, see the upregulation of all the suspects that are downstream of this signaling pathway. They're not the same between Orphate and IL-17. They're similar but distinct, right? Mm-hmm. So what Orphate induces is similar to what IL-17 induces in terms of the cytokines, but they're not exactly the same, which is not surprising, right? Because they're slightly different proteins.
Right.
But what is emerging is that apparently Orphate, as Amy said, is a mimic of IL-17. It binds its receptor and turns on the same signaling pathway.
Well, it turns on but that leads to 3 of the same critical components of the pathway.
Yeah.
IL-6 and CCL20.
Now, this may be a good time to ask this instead of waiting, which I would normally do. What would be the function of SARS-CoV-2 of turning on a pro-inflammatory pathway, which would clear infection in theory, right?
I don't know. Maybe if you clear infection, it helps you cough it up and transmit. I don't know.
That's too anthropomorphic.
Yeah, I don't know what it's. I don't know.
But you see the conundrum I have, right? That's
right. But the other thing is I don't know when it's turned on. How late in infection is the virus still replicating, or is it finished? You know.
Well, when is Orphate made?
I'm not
Should
sure.
be made early in infection with everything else.
I'm not sure because it's from a subgenomic mRNA.
Okay. But just having this activity.
Yeah, I'm not sure.
It's weird. But Orphate, you say, does other things as well, right?
Yeah, it also, impedes T cell migration and stuff.
Right. So maybe this is a side consequence that you can't avoid. And
aren't some of these pro-inflammatory cytokines required for T cell? So, or prevent T cell migration or something?
I don't know.
That's
a good question.
I mean, you might turn it on because you want it to turn something else off. I don't know.
All right. Okay, so then they ask, let's learn about the receptor that's involved in binding Orphate. And they say, as we said, IL-17RA, the receptor A, can make a heterodimer with B or C. And they want to know what Orphate is binding to. So they have cells that produce different combinations of IL-17 receptors. And they find that A and C receptor have the highest binding to Orphate. It will also bind to others, but that's the highest, A and C. And then they wanted to know how the receptor complex affects downstream signaling and inducing, phosphorylation of NF-kappa B, the production of cytokines and so forth. So they they knock out each receptor subunit individually in these cultured cells using CRISPR. And then they add Orphate and they measure the activation of NF-kappa B. And they find that when you knock out IL-17RA or C, you really decrease the signaling after adding Orphate protein. But IL-17RB, not so much. Doesn't care. Yeah. So IL-17RB doesn't seem to be involved in Orphate binding.
Nope.
Okay.
Mm-hmm.
So basically what we have here is that Orphate, in fact, Orphate induces the heterodimerization of the receptor. So the receptors, without a ligand, they exist as monomers. And then when the IL-17 binds, then the other subunit comes, and then that triggers that heterodimerization, which is ligand-induced, triggers the downstream events. And they find that, in fact, Orphate does the same thing. Mm-hmm. It binds one of the receptors A, and then C comes along, hydrodimer, and now it activates downstream signaling to get your inflammatory responses.
Yeah.
You're all good so far?
I'm good.
So then they make a lot of changes in Orphate to ask, can we make an amino acid change that would decrease binding to the receptor? And they make 12 different amino acid changes, and they find 4 single amino acid changes that have decreased binding to the receptor on cells. And these are 42, 106, 71, and 76. And what they find is that 42 and 106 and 171 and 176 are critical for the interaction with the receptor. Those 4 amino acids based on their kind of limited mutagenesis. Mm-hmm. Now, in people, who are infected, they often contain variants of Orphate, and those variants are observed at position 24, 62, and 84.
Yeah. And like 84 was identified really early on during the pandemic.
Right. I remember that. Yeah.
It was like identified unbelievably early on. So almost every single virus has an L84S alteration.
Is that so? Today, almost every virus circulating has 84.
Mm-hmm.
Interesting. Anyway, so those 3 amino acids are not the same as the ones they identified by their changes. But so they take these 3 amino acid changes and put them into Orphate, and they ask what's the effect on binding to cells. And then, Amy, guess which change had the most effect on binding to cells?
L84S.
L84S, indeed.
Yeah.
It really knocks down surface binding of Orphate to cells.
And no IL-6 and CCL20 induction.
That's right. And there's no CCL made. Oh, Amy, CCL20 is the cytokine we looked at in the previous paper, isn't it?
Yes.
I just realized. I just had a moment of
clarity.
Thank you.
Yeah.
CCL20. Yeah. So in the other paper, it was downstream of a junk
PK,
pathway, right?
Yeah. But that's probably regulated by the receptor.
Yes.
I'm sure that off of the IL-17 receptor, there's the J and K pathway.
So L84S makes less pro-inflammatory cytokines. And as we said earlier, it's associated with milder disease.
Uh-huh.
So is it fair to say that anybody who gets severe COVID doesn't have an L84S deletion, or we don't know?
We don't know.
Okay.
I wouldn't say that. There could be other alterations that compensate for the absence of Orphate function.
Amy, if you were, here's a loaded question. If you were going to make a attenuated vaccine, would you delete Orphate?
No, really? Loaded? Would that be a grant that I wrote?
That's exactly right. That's why I called it loaded. Yes.
Yeah. Yeah. And somebody else wrote a similar proposal. Yeah. I mean, yeah. So that would be a starting point. There'd be one change that you'd want right away because you don't want to induce all these pro-inflammatory cytokines. Yeah. Right. But I mean, we were going to combine it with like a deletion of ORF3, which is an interferon antagonist deletion.
Right.
Yeah.
That's it. Those are the data. But
yeah,
I think
it's really cool.
It's interesting. So IL-6, Orphate is an IL-17 mimic and binds the receptor, induces the synthesis of pro-inflammatory cytokines, which probably are playing a role in the cytokine syndrome, severe COVID. But then, a naturally occurring single amino acid change in
that occurred
Orphate
early on in the outbreak.
Reduces binding to the receptor and reduces the production of these pro-inflammatory cytokines. Now, nevertheless, Amy, a lot of people have still died.
Yeah, but your assumption is that they all died because of a virus, and they didn't have any SNP in any protein or anything that would have altered the course of pathogenesis.
So that's the question. And I asked you it already, but do we know if everyone who dies has an intact Orphate? We don't know that, right?
We don't know, but even still, did they all have an intact, IL-16 or IL-6 gene, or did they have a gene that, or that was hyperactive or something or other?
Oh, right? You're saying
you don't have any host factors. You're saying
okay. So you're saying host factors could compensate for viral.
Yeah, they could compensate.
Changes, yeah.
Plus, the other thing is even if you all had L84S and Orphate, there could be disruptions in Orf3B, which doesn't antagonize interferon or over antagonizes interferon, right?
Yes.
So you could die because you had a change in ORF3 that made it super potent, right? So, I mean, it's not clear to me that it's a one gene phenotype. I don't think death is a one gene phenotype.
I'm sure someone has looked at, Orphate changes stratified with disease severity, right? Not just
they
that.
say that some of these correlated more with disease, with mild disease, right?
Yeah.
But
that's some. It was a limited study. I'm interested in a more extensive study.
Well, I'm sure if you go to your Covavax or whatever the website is,
covariance.org,
covariance, you can figure out how many that you'll see that the majority of them have an L84S alteration.
Let's look.
Okay, go ahead.
The thing is, though, I don't think they just look at individual lineages. Can we look at mutation? Oh, here, mutation. You can select by mutations, and they're all mostly in spike, and there's one in ORF1A. So they're not following ORF8. Kind of crappy, don't you think?
I'm not in charge.
Well, you should be.
No, I have other stuff I'm in charge of.
View shared mutations.
Charge of.
So here's another way of looking at it. Shared mutations.
Okay. Tell me what shared mutations say.
Ah, boy. Spike, 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 spike. They're all spike. They're all bloody spike. It's all people focus on is bloody spike.
Sorry.
I'm sure there are proposed changes to this section. I should propose changes. How about some other mutations for God's sake?
Let's just look at the variants of concern. I'm sure the genetic thing, the alterations of the variants of concern are listed next to the variants.
Well, yes. Here's defining mutation of BA1, for example.
Okay,
Spike,
so it's like 60.
Spike, nucleocapsid, ORF1A, 1B, 9B, E, and M. Nothing in ORF8.
That's surprising.
So that's BA1. How about BA2? Can we find bloody BA2? Let's see. No, they just call it Omicron. They're all Omicron, so they're not doing a BA2. And you can't search. When you need something, when you first find these lovely pages, you say, oh, that's cool. But then when you really need to find something, you can't.
So what you're saying is you're just seduced by the aesthetics.
You
like
said
the
it.
Pretty woman who walked down the street.
No, I'm not saying it that much. I'm just saying the pretty nucleotides all in a row, you can't find what you want. And now I'm sure if someone's going to write and say, if you knew what you were doing, you could find it. Well, fine, find it for me. Let us know. I want to know the ORF8 frequency in severe versus non-severe COVID. Now, as Amy would say, it's not just one thing, Vincent. It's multiple things. I understand that, but I want to see how this stratifies. It's an interesting observation for sure.
Well, yeah, I mean, it's interesting, and it's, it gives it another aspect to the disease that we don't necessarily focus in on. All
right, one more thing, Amy, here. The last paragraph, they summarize what they find, and they make a statement that rubs me the wrong way.
Okay, I'm sitting down for this.
They say, being a hotspot for variation, there is always a possibility that a new ORF8 variant emerges that has acquired enhanced virulence. Why would that be? Why do you think it would have enhanced virulence? I'm
not sure. Well, because maybe what they're saying, so it's also a hotspot for deletion, right? And so by analogy, if you said the loss of ORF8 attenuates the disease, then I guess the deletion. Well, no, that wouldn't work out.
No, the thing is,
I don't know why you would write that.
Maybe you could have a change in ORF8 that enhances his activity for whatever function it's needed, and just as a side effect increases the virulence. Maybe it improves binding and signaling and in pro-inflammatory cytokine production. That's not the primary selected phenotype. It was something else. So in other words, increased virulence could be an accident of something else happening. But on its own, it's not a selection, right?
Right. It's an accident.
All right. It just bothered me because they just throw it out there and they don't really explain.
I don't know why they threw it out there. I don't know why it's not explained. But I also don't know why the assumption is always it's going to evolve more greater virulence.
Always, that's always, and when you think about it, anyway, I've talked about it enough. Okay. I want to read a couple of emails here.
Okay, we can do some email. Is that what you're saying? You want to do some email?
Yes, I am saying.
Okay.
Can you take the first one, please, from Colin?
Okay, Colin writes, I'm a big fan of the show and have been lucky enough to have Vincent, Brienne, and Alan Dove as guests on my show at various times. Oh, this is the guy I think is related to Johnny Mac, right?
I don't think he's related. It's
well,
Colin
not,
McEnroe.
But every time you say you're going on the Colin McEnroe show, I say, Johnny Mac?
Yeah, that's right. That's right.
I've been a little puzzled by the somewhat laissez-faire attitude towards masks, given the serious sequelae associated with even mild symptoms of COVID. Aren't we rolling the dice if we get lax about pre-exposure strategies? Thanks for all you do. I'm going to yield the balance of my time to the great Laurie Garrett. Here's the bottom line. You don't want to get hashtag COVID-19, period. It will be less dreadful if you're fully vaxxed, but the virus damages the immune, cardiovascular, and neurologic systems, deregulates the pancreas, kills neurons, directly damages the heart. Be safe. Be smart, be safe. Colin McEnroe.
This bothers me to no end, this email, okay?
I figured that's why it was here, and I was supposed to read it, because if you read it, you'd be too annoyed.
What are your thoughts? What are your thoughts, Amy? I'll just be quiet for a bit.
What are my... No, you don't need to be quiet. You can...
What do you I want to know what you think about this idea that you should wear a mask because you might get long COVID.
So if you're vaccinated, it's not clear to me that there's a large percentage of people who get long COVID.
Yes, I agree.
Okay. So I'm not saying that if you're more comfortable, you shouldn't wear a mask. I'm just not sure I need a mask mandate. I think, you're pretty smart and most people can figure out. So, yeah, I'm not clear that there's more long COVID.
I agree with that.
So the way that she states it is the virus damages the immune, cardiovascular, and neurologic systems and deregulates the pancreas. So that implies direct viral damage. And I'm not clear that there's direct viral damage in the nervous system or as a consequence of, not being able to have a large enough tidal capacity volume to breathe in, and you have hypoxia, and that doesn't occur.
Yeah.
And that would occur versus like severe flu. If you got a severe case of flu, I would imagine some patients develop hypoxia.
Yeah.
So I'm not sure that that is correct. I don't know the percentage of people who develop cardiovascular disease from COVID, but you can get cardiovascular myocarditis and stuff from enterovirus infections, ECHO1, Coxsackie bees. So I don't see anybody looking into that. If they do, you should give me some money because that's been the focus of our research.
So the way I look at it, I agree with that. And there's no evidence that the virus is directly doing this. I think it's a inflammatory hypoxia consequence. Virus does not kill neurons. I'm sorry, this is just not right. You may think Laurie Garrett is great and she's very wonderful in many ways, but this is wrong for the most part.
Yeah, I think she's great for certain things that she advocates. I think she's great at advocating for a unified public health policy, having public health officials in charge of making public health policies and stuff. I'm not necessarily sure she's a great MD. or virologist, but you don't they're different. You need different skills for different things. And I think she has a great set of skills for advocating and pointing out the holes of why you should not have trained MDs and not public health officials running, the CDC and the city health departments.
Yeah.
I think that that's great. And I think she has a great platform for why we should have universal vaccination and the economics of, vaccinating the world. Like if you just look at the bill that they're trying to pass for more COVID funding, right? They're they're taking out $5 billion, right?
Yeah.
And where's that $5 billion from? And $5 billion is the $5 billion that was going to underwrite our efforts to vaccinate internationally.
Of course.
Right?
Of course.
So she's great for addressing those kind of questions, right? That's what she's great at, right?
So what you're saying is stay in your lane.
Yeah, but I'm not Daniel, so I can't do that because then somebody will write in and say she's an arrogant piece of shit.
Actually, I was just going to say that you were being very measured today. I mean, in your description of Lori, you were very measured, which is great. It's very good.
I don't personally know her, but I know that you and Ian have had great interactions with her and are very fond of her, and I trust your opinions. And so, she's a close friend of Ian's, right?
Yeah.
When she's not falling out of kayaks.
No, but this statement of hers is completely inflammatory and wrong. If you're fully vaxxed, it's not just less dreadful. It is much less dreadful, and you're not going to damage your immune, cardiovascular, neurological system, blah, 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 after you're vaccinated. It simply doesn't happen after you're vaccinated. Okay? Right? Wouldn't you agree?
Right. It's generally an abortive, for the majority, for 90% of the people, being fully vaccinated and then following it up with an infection is generally an abortive infection, right?
Exactly.
You make a little bit of virus for 24 hours and then it's done, right? Basically.
Yes,
I mean, we're simplifying it, but that's pretty much the way it works for 90% of the population, right?
Yeah, yes, right. And that's why, Colin, we are not being laissez-faire about masks. We're basing it on the science.
Well, I'm not being laissez-faire. I mean, laissez-faire is a free marketplace, right? And so at this point in time, I think we all have all of the knowledge that you need to make a decision. And I am going to trust my fellow man to make the right decision. And if I'm not going to trust my fellow man to make the right decision, then I personally am going to wear a mask. I go into the grocery store, go to Zabar's, I wear a mask, not because it's mandated, but just because, maybe there's some kook out there.
But you're fully vaccinated. You're protected, dude.
I know. But sometimes,
All right. Listen, Colin,
you
get
wear a
Amy
mask on the train, right?
Well, I have to. If I didn't have to, I wouldn't wear it.
Right.
I don't wear it where I don't have to any longer.
Yeah.
Because I'm vaccinated and and 90% is good enough for me.
Yeah. I think it's all good.
When you drive, the chance of you dying is far more than 10%.
Well, yeah, the rate that I was in the car on some super highway in Maryland, and I was not driving the vehicle, but the vehicle driver kept pointing. So every time a car almost cut him off when they were switching lanes, which they did every 5 minutes or less, and they did so without their signal, he would point it out to me. He would say, there it goes. See it weaving? There it goes. It was like watching a tennis match. I was getting sick. I was like, can we pull over? No, I'm not so excited to drive the car.
So, Colin, you should have Amy on your show, okay?
No, I don't need. I have other jobs.
Bob writes, hi, #myheroes. Well, thank you so much. Here in Simcoe County, just north of Toronto, it's Monday morning, -4°C, sunny, calm, with a good bit of snow from yesterday still on the ground. Why am I already nostalgic for snow? It's already warm here.
Because you're delusional.
For what it's worth, I made the attached for a teacher friend who was distraught yesterday, relating to me about the great losses she sees in her high school students. She reported a general loss of inner confidence in the teens with profound doubts that they are competent to move forward in the world. She also noted a sharp dichotomy of similarly scary presentations in her school. Overachievers who can't be satisfied with A's or even A pluses, and completely non-functioning students with minuscule amounts of coursework completed. I had little to offer my friend at this time except a realistic look at the numbers over the past 2 years. The math is particularly easy right now, with average daily death rates essentially the same as they were 2 years ago at this time. Then, of course, case rates and death rates were going up. Now case rates are at least 30 times higher than they were in late March 2020, and who knows which way they are going anymore. I'm more confident the death rates are a bit more accurate, or at least that the trend direction is going down. Biggest thing I take away from the attached is this is what we would have hoped to see about now if our intent in March 2020 was to flatten the curve. Our docs now have tools in their toolkit, but they and teachers are exhausted. Time for love and respect from any of us who are able to keep things a bit safer, which I have no doubt you all continue to do effectively for thousands throughout the world, Bob. And what he sent was a kind of a graph, global COVID case fatality rate 2020-2022, which just goes 10% in late March 2020, late March 2021, 2%, and now less than 0.3%. And this is from a website from the 7 day at worldometers.info. And so, yes, I understand that. So what he's saying is that a lot of the loss of inner confidence comes from the pandemic, do you think? But I'm not sure that teens really know about death rates, do they?
I think so, yeah, for sure.
Yeah.
Yeah, I think so. I also think that the loss of inner confidence is not just death rates. It's the whole social upheaval that occurred.
That's why I think we need to stop wearing masks and get back to life, because this is now under control and you got to take care of this other stuff. I totally agree with this.
Yeah, I think it's the right thing to do. I agree. I also, I'm not 100% clear that, where now case rates are at least 30 times higher than they were in late March 2020. I don't know where he got that. And who knows which way they're going. I'm more confident that death rates are more accurate. Why, you die faster now than you did in 2020?
No, so the problem with the rates is that the testing really varies, right? So you never know. We're just testing a lot, and
yeah, I'm just a little confused. I mean, I think the point is that it's becoming less trauma, and you're able to go to school and have social interactions, and at some point, you're going to have to trust. We're going to go back to the way life it was, and we're going to trust the person who's next to us to have, stay home when they have the sniffles. And you can't always assume that the sniffles are now SARS-CoV-2 infections versus influenza, and we had no problem.
Yeah.
When it was an influenza pandemic or an influenza epidemic or outbreak about not masking and returning.
Can you take the next one, please?
Sure. Melanie writes, "Hi, Vincent. It's a cold day in Ottawa, currently -7 with a wind chill of -17. Yeah, I'm not reminiscent about these days. I don't need any more snow. But yeah, no more snow. I was wondering if you could find out about the COVID-19 vaccines that are not intermuscular. Are they all live attenuated? And if so, why? I have read that the live ones are needed to enter the mucosa. Could a benign viral vector work? Or is it that I don't have a clue about the technology? I feel that more people would get vaccinated if they didn't have to face a needle, but I do not know how uncomfortable it is to get a nasal spray or inhaled vaccine. I just assume it would be relatively easy. A topical would be the easiest, but that's probably ridiculously hard, impossible to make effective. And I'm sure that the scientific community has considered pills and is also too tricky. Also, do you know anyone using artificial intelligence to find ways to improve vaccines? Is there any hope that a neutralizing vaccine could be developed for COVID? What I mean is that a vaccine be developed that could prevent shedding to infected others. I don't know if that's the true definition of neutralizing. Sorry for so many questions. You and your team have guided me and kept me calm during these. You have tumultuous times, Melanie. So I believe that Michael Diamond and various other people have tested the mRNA vaccines intranasally in animal models and that they've seen that they can work. But I don't know that they have gone any further into people. Not sure where it stands.
Remember that we did a paper with I think it was an intranasal vaccine with 3 different viral proteins, but they were adenovirus vectored, right?
Yes, they were adenovirus vectored. But I believe that Diamond has looked at the mRNA vaccines intranasally.
And that worked? That gave protection?
I think it gave protection in a Syrian hamster model. But I don't know where it stands. And I don't know, we've talked about making microneedle patches like for polio, like from the guy at Emory. And I don't know where that stands at all. I don't follow it enough.
So Melanie, microneedle patches are these, what they say, you put them on your skin with a Band-Aid and they deliver the antigen to your dermis. I think they're going to be replacing needles in the next 10 years because they've been being tested a lot and they work very well. They're quite immunogenic. So even though you think the topical would be hard, impossible to make, in fact, it's under development, as Amy says. Yeah.
I think a pill is not a good plan because many things degrade in the pH of the stomach.
Well, there was a adenovirus vectored vaccine given in an enteric coated capsule that would get through the stomach and then uncoat in the intestine and deliver the virus there. I think there's a COVID vaccine in early testing, but it hasn't gotten very far, I think.
Yeah,
so that would be a mucosal immunization, and then it would also provide mucosal immunity in the lung, right?
From the gut?
Yeah.
I don't know that you would get mucosal immunity from the lung from a gut.
Yeah, sometimes when you immunize,
sure.
When you immunize one mucosal site, it goes to another.
Sometimes, I mean, there's a lot of stuff that happens in the gut that influences lungs. Like leaky gut is what leads you to lung failure. Like when you're a burn victim and you have leaky gut, all that commensal bacteria then moves into your lung and gives you sepsis.
Yes.
So, I mean, definitely gut health influences stuff, but I don't know. I don't know if you get IgA or just IgM or IgG. I mean, I don't know.
What do you think about AI to improve vaccines, Amy?
Sure. Go ahead. Try it. However, I don't think you're ever going to make a neutralizing vaccine. We've never made a neutralizing vaccine.
That prevents shedding?
Yeah. I don't see how you're going to block infection.
It would have to be sterilizing, right?
Yeah, it would have to block infection, and we've never made a vaccine that blocks infection. I don't know how you would do it.
I agree.
I don't think that's ever going to occur.
Josh writes, I loved your podcast episode about the monoclonal antibody offering protection against hantaviruses and the oral remdesivir-like antiviral. I had a question regarding the first paper featured in that episode in your discussion of pseudovirus use in research. You mentioned that in this study, the glycoprotein arrangement on the pseudovirus may be different than on the true hantavirus, possibly explaining the discrepancy in the antibody's neutralization capacity, pseudotype versus authentic virus. Why are pseudoviruses still used so often in virology research, despite the potential for such discrepancies? How reliable are results from studies involving pseudoviruses opposed to authentic virus? And does this vary by the type of virus? Thank you so much for your time, and thanks for the great podcast. So I think this is a good question.
I think that this is a pet peeve of mine.
Yes. So why don't you address it, Amy? What are your thoughts?
Well, I think that pseudovirus I think it's a good tool, but I think it's way too heavily relied upon. I think that there are conclusions that are not correct. And I think we automatically assume the easiest assumption is that when you pseudotype something, that the protein is actually in the proper orientation, and then whatever you're looking at, like the antibodies, will be neutralizing against intact virus and be a prophylactic. And actually, I just asked somebody that yesterday because the vaccine for Ebola is pseudotyped VSV.
Right.
And so they talk about how they pull out all these antibodies that are elicited against the vaccine, right?
Yes.
From patients. And I asked him whether or not they were the same epitopes that were known to be identified as neutralizing epitopes from natural infection. And the guy just looked at me like, what? Not so good. And so, yeah, I think the lesson of HIV should have been retained longer. So we know that when we pseudotyped virus and produced it in 293 T cells, GP120 and 40 had a different repertoire of antibodies. And we tried to use those antibodies as prophylactics against virus infection, and all of them basically failed.
Yeah.
And so I think you have to, in order to use pseudotypes, you have to well characterize them before you do any assays with them and make biological conclusions. And I'm not sure that that's always done because those assays can be expensive, like the cryo-EM structure of the pseudotype. Has there been a cryo-EM structure of spike on top of VSV? Or are we only looking at spike in the context of the virion?
Well, it's just the protein, right?
Is it just the protein?
When McClellan did the spike structure, how was that? Let's look it up.
Look it up.
Spike. Cryo-EM spike structure of the SARS-CoV-2 spike in the prefusion conformation. All right. So basically, we expressed ectodomain. They just did the protein. Yeah.
Yeah.
I think it would be interesting to do it in the context of the membrane for sure. And I would think SARS-CoV-2 versus pseudotype would be very interesting.
Yeah. And all of our neutralization assays where we come out and say, oh, this variant is less sensitive to our monoclonal therapy. Maybe they're wrong, or there's not exactly the right conclusion.
Could be. I agree with you. So the problem is, as you know, Amy, you have to do authentic virus, SARS-CoV-2, at least at BSL-3, and not everybody has that, right?
Yeah, but the labs that are at the forefront, they all have a BSL-3.
They do. But you know what the problem is?
I'm waiting.
You got to line up for the BSL-3 so you can do your
downstairs. Downstairs, it's basically a SARS-CoV-2 room.
So then why don't they just do SARS-CoV-2?
I don't know. But we know somebody who wanted to use the BSL-3 and was for a different virus and was told that they could not.
Yeah.
Or at least I know of that person because he came to talk to me the other week about some gossip.
But, the problem is that
I think it's easiest and fastest if you're just able to crank it out with a pseudotype virus, so you don't want to actually go the extra yard.
Some people say they can't plaque SARS-CoV-2. I know a very close lab to us who says they can't plaque SARS-CoV-2.
Yeah, but now they figured it out.
Oh, yeah? What was the key?
The key was exactly what I suggested years ago, was that their monolayer was too confluent. And so instead of looking at plaquing on a 90% confluent monolayer, I believe they're now plaquing on a 60% confluent monolayer. But
see, people don't want to listen to you, Amy, for whatever reason, and then they waste time and finally come around and that you were right. So,
well, they didn't exactly say that I was right. That's not what the lab does. They don't do that.
Well, if we did more of that in science, it would be better. Oh, you were right. Thank you so much. It would really go a long way.
It would, but, people
well.
don't like to be wrong. And they definitely don't like to be pointed out that.
But I do think it's easier to do a pseudotype than SARS-CoV-2. Okay?
Yeah, I mean, gowning up is just another 20 minutes of drama.
Yeah, I mean...
Let me tell you, Vincent, it's another 20 minutes of drama, okay?
I'm sure it's drama. I mean, I saw you put on the helmet once and you freaked out.
Yeah, and then I had to put on the suit, and then you can, you put on the suit, and then you put on all this other crap. And by that time, like, the belt for the headgear is too heavy, and you can't, go like this with your arms because you got too much other crap on. I mean, it's a pain in the butt.
All right, there's one more. Let's do this last one from Simon. I always have to thank you for helping me keep my sanity through this pandemic. Now more than ever, and I and others need your help. At one and the same time, we have anti-vaxxers digging in. We have governments acting as if there is no pandemic at all. We have public health officials and epidemiologists talking about waning immunity and Omicron BA2 being as infectious as measles. Nope. And we have a small group saying that everyone should be mandated to wear N95 masks all the time and that vaccines are effectively useless even though we should use them. As it stands today on social media, I'm as likely to be blocked for being a let-it-rip anti-mask scandemic person as be blocked for being anti-lab leak and pro-vaccine. So what can scientists do to bring some rational calm when we have madness on all sides from politicians, public health officials, and medical professionals? Like our politics, thanks for avoiding. Notice he thanks us for avoiding politics. I regularly get emails saying, stay away from politics. We don't do politics anymore. Okay? Simon gets it. Like our politics, the battle we have is to bring people from all extremes back to the rational middle ground where we can debate what the latest data actually means calmly and with nuance. Yes, that's the way to do it. And probably me getting excited doesn't help, but this is a limited audience. TWiV does this, but this morning I was blocked by a pregnant woman with under 5-year-old children and a friend with MS who was living in fear and treating me like I was Kennedy, Wakefield, and Malone combined. This morning I saw a professional doctor tweeting about being in fear when they go to the grocery store. Thank you and TWiV and Microbe TV for everything you do. I value the discussion. I value the calm debate. I value the honest disagreements and discussions across all of your podcasts. Keep up the great work you do.
Maybe they should sit in on one of our discussions, that it's not so calm.
Yeah, but it's between you and me, Amy. The world doesn't get to see that.
I know. I'm just teasing. Well, periodically a few people get to see it, but
Simon, your point on, it's very difficult, and we encounter the same here. We try and be calm, but I do think your point here, we have to go to the middle ground and have a calm discussion with nuance is the key. But many of these individuals, you can't have a calm discussion with. So.
Well, many of these individuals think that they entrench themselves in their point of view. It takes a really big person and a really open mind to acknowledge that maybe they didn't know everything and that they're flawed. And they, as Daniel and you like to say, they are out of their lane. And for various reasons, people go out of their lane. I mean, scientists really go out because that's where the money is, right?
Yes, absolutely.
And if it were more equally distributed, there wouldn't be so many who go out of their lane. But then you would argue that maybe you don't get all the different points of view, and science wouldn't be as sciencey as it is without all of the different points of view and input, right?
Yeah.
But I think we also have to accept that things change and we've made mistakes, and it would be better if everybody took the philosophy of we did as well as we could with the knowledge that we had today. If you come back and tell me tomorrow that I miscalculated, I'll be happy to revise my hypothesis. But today it's very black and white. You don't have that ability to walk it back, and you should. You can start off your walking it back with, I did the best I could. I'm sorry I made this mistake. I didn't know about this piece of data, or, whatever it is.
I agree. That's a great way to do it. Yep. Nobody does it.
No.
We try.
Yeah. I mean, but I wouldn't say nobody does it. They do it in a non-public fashion. You can, Edmund and various other people and other people that I've had conversations with about just topics that I do my own research on. They'll be like, Amy, what about this? And I'll be like, oh yeah, maybe you're right. I didn't know about that piece of data, right? Or something or other. Or I didn't count that factor in, or that's great. I didn't see, we made more progress. So yeah.
Yeah.
I mean, but it occurs, quietly and in the privacy of our own lab, right? It's not out in the public. I'm not a public figure.
I agree. All right, Amy, let's do some picks. What do you have for us this week?
I have the Ken Burns 4-hour documentary about Ben Franklin. So not just that fact that Ben Franklin actually was the most famous American at his time. Nobody knew about anybody else, especially in Europe. They all knew about Ben Franklin now. But it turns out that, during his lifetime, during the Revolutionary era and stuff, there was a pandemic of smallpox, and his son died of smallpox. And there was a inoculation mandate. So this was slightly before Jenner's era. And so Washington had an inoculation mandate that if you wanted to come and fight for the Revolutionary Army, you had to be inoculated. And so we've had this debate in this country about inoculations or vaccination and mandates since before we were even a country. So it's kind of disconcerting that we still can't get past it or find a resolution where, we can have civil debates or, whatever about it. So I think it's interesting, but it was a really good, it's being released. It was, and Ken Burns makes great documentaries.
How do you get to see it?
So I read about it and I heard about it, and he's been interviewed about it, and he points out and you can see some trailers and stuff, and he points out about this episode. And then I did a little bit of research about whether or not Washington really had this inoculation mandate and stuff, and he did.
I'd like to watch it, but I don't know how I can do it. I have to figure it out.
What do you mean you don't know how to watch it?
Well, it says you have to pay for it.
Oh, really?
Yeah,
At some point it'll be on PBS.
What's that? Just kidding. I go to the page and it's got a description of it, and then it says stream our films. So I click on stream our films, and it brings me to Amazon. And it says start a 7-day trial. It's on PBS, but you have to have it through Amazon. So anyway, I have to figure it out. In contrast to this, all of our films are free. Everything we do is free, folks. Yeah, I know. Ken Burns has to make a living. He's got to pay everybody. I get it. But there's some things people need to know, and that's why we give it to you for free. You need to know about viruses.
Yeah, he's made some interesting documentaries. I don't know that I think my former brother-in-law was involved in it. The documentary about the history of the basketball, about the NBA. It's interesting, sure. From a social perspective, do I actually need to know it?
No, you don't need to know.
Probably not.
But
I'm not a big basketball fan.
If you're interested, if you want to be entertained, I think it's more important to know about viruses, frankly.
Yeah, but he has some other things that are really interesting.
In fact, I think every college student in the country needs a course on viruses.
Well,
Certainly.
every university should have a virology course or have it included in basic biology.
So at Columbia, the first year students take a course called Frontiers in Science because it was decided a number of years ago and decided correctly that all students, no matter what their major, need to know a little bit about science. And one of the courses, or one of the sections, is about astronomy. There's no virology, there's no microbiology. Now tell me, how is astronomy going to be important for your life as opposed to virology or microbiology? Well, it's not. And in fact, I've had discussions with Columbia students who will remain unnamed, and they said, yeah, it's more like it'll make us better at cocktail parties.
Would that be my undergraduate?
No, he didn't say that to me.
That does sound like something.
Does it sound like him?
Yes,
But
it does.
come on, folks, get rid of the astronomy. I know astronomy is cool, but it's not going to save your life. If you know about viruses and microbes, it could save your life one day. And anyway,
yeah, but at the end of the day, the only thing that's going to save your life
get
that
vaccinated.
you're in charge of is swimming. No, I'm serious.
Yeah, you need to learn how to swim, but you should know that before you go to college, right?
No, there's a lot of, you'd be surprised that there's a lot of people who don't know how to swim.
No, I agree with you, but I'm
And
talking
like,
about a
I believe
course.
that there's actually a swimming mandate at Columbia, like there is at Emory.
That's
And
fine. That's fine.
Is
I'm
that
just
you-
talking about a course where you sit in a class and learn stuff.
Yeah,
I
I
agree with you 100%, but I think Columbia students should all have some virology, maybe not a whole course, but a bunch of lectures from me. And I think it should
I can't believe that Moshowitz does not have that included.
it's not in the Frontiers in Science. And frankly, I think astronomy is absurd to have in there. I'll argue with anyone to the end of the day, to the end of the earth, to replace astronomy with virology. It's absurd.
But
Ridiculous.
wait,
What?
but Masha's class is not the Frontiers of Science, right?
No, Frontiers is a mishmash of different subjects team taught, all put together in the first year. And it was put together by committee, which is always a problem, right?
Well, they have their own pet projects, right?
Yeah,
But
exactly right.
But you've known Moshowitz's for like 3,000 years.
Yeah.
So why aren't you part of her class?
No, I don't need to be part of her class is biology. That's separate from Frontiers in Science. Okay. I'm just talking about the thing that her class is mainly taken by bio majors, right? Or people who want to be people who want to do some science. But, if you're a history major, you don't need to take her class, but you have to take Frontiers in Science, which I think is fine. But I don't think you need astronomy. I'm sorry.
Oh, yeah, no, probably not.
All right. I have a pick, Amy.
I know. I'm waiting since they're articles that I sent because
Well, one of
I
them
was
is
either outraged or agreed with or something or other about. I'm always interested to find out how I influence people.
So I have two picks, and they're both about the second booster dose that was recently authorized by the FDA for 50 and up and immunocompromised. Okay. And
sit it down.
And so when I first saw this, I went and looked at the Israel data on which the decision was based. And I said, no, this doesn't make any sense. And then Amy sent me this article from the Wall Street Journal by Philip Krauss and Luciana Borio, people who were formerly associated with the FDA. So they know what they're talking about. You likely don't need a fourth COVID shot. Unless you're at high risk, the initial 2 doses are enough. And 2, not even a first booster, they're arguing. And what is the crux of their argument? 2-dose regimen is enough to provide most patients excellent protection against severe disease mediated by durable cellular responses, not the neutralizing antibodies that rise and drop quickly after vaccination. I mean, boy, they put exactly what we've been arguing for the past year into one sentence, and it's perfect. The New York State Health Department's large database shows the effectiveness of full 2 mRNA doses remained above 90% against hospitalization, including during the recent Omicron surge. A study from Sweden found the same. Studies in Cotter in California show no decline in protection against severe disease with Omicron. And so that goes on. And So the Israeli study, a fourth dose reduced the likelihood of severe disease, but the effect was driven by those with major risk factors. There's no evidence that introducing boosters last fall had a significant impact on the course of the vaccine. So unfortunately, the Wall Street Journal is paywalled, so you might not be able to read that. But my other pick is...
what are you talking about? I sent you the PDF. You just slap it into the thingy.
Oh, they'll go after me. They'll sue me left and right. I don't want to be sued by the bloody Wall Street Journal. But if someone wants it, let me know. We might be able to arrange something. And
what are we doing? Back alley trades now here?
I'm trying to be subtle and you call me out, man. All right, ABC News. With fourth COVID vaccine doses looming, experts say not so fast. You know, this is Paul Orphate is here quoted. Well, first of all, Anna Durbin, who is at Johns Hopkins, she developed a dengue vaccine. There are very few, if any, people who, in my opinion, require a fourth dose. Paul Gopfert, general, it's too early to recommend a fourth dose, except for those who are immune compromised. And I would say even you're immune compromised, you may have 10 doses and never respond. You should just have antivirals and monoclonals at your disposal through your physician and be ready when you get sick, for God's. I'm not sure any number of vaccinations is going to help you. But where's Paul Orphate? Unless there's clear evidence something is of value, don't give it, said Paul Orphate. And Paul Orphate basically said that the Israeli data are flawed. I agree that they're flawed. He said they're flawed because they use people who sought a fourth dose. And I think they're flawed because they didn't distinguish. So they compared people with one booster and two boosters in terms of death rates in different age groups, but they didn't stratify them according to other variables that might affect the outcome. And the numbers are very small, and I don't think we should base our public health policy on that. And so it's not just me arguing that. Daniel agrees. Borio, Krauss, Orphate, and many others. You don't need a fourth dose. You don't need a second booster. The science doesn't say it. And these two articles will give you a flavor for that.
Yeah,
Amy, we got some pushback on the live stream the other night from people who said, well, is it any harm? What's the harm in taking it? There's no harm, but you don't need it.
Yeah, well, you know, look, there, I appreciate the people. I appreciate the fact that there's a significant population that trusts science and trusts the government, and that, you know, when it comes down to, you know, your health and various other things, there are a lot of things that, you know, people pay for that you probably don't really need. And the argument is, well, if the government pays for it, obviously it's good. And I'm like, okay. But, you know, I don't necessarily want to throw out the baby with the bathwater. So
get 2 doses. If you want, you can get a third dose. You don't need 4.
Well,
Sorry.
you don't need 4. And I think that there's a large amount of stress on antibody levels by the scientific community. And those people, unfortunately, get a lot of coverage, and they have large Twitter presence and stuff. And so that you then have a large group of people who say, but, you know, person X on Twitter is all about the antibodies so that they may be correct. Whereas your candidate today said, yeah, we're all about the antibodies, and that's not correct.
So Daniel said, we can't even get 40% of the U.S. 1 dose. We should be working on that, not giving the rest a 4th dose. I like that. Well,
that's what Paul Orphate has always said.
Yeah.
Paul Orphate has come. You should have the lady who is from Philadelphia out of Temple. I sent you her information. That's Paul Orphate's colleague, or he knows of her. Maybe he can help you get her on TWiV. But she goes out to the vaccine hesitant in North Philadelphia, and she calmly talks to them. And she even sometimes, I believe, takes them to the vaccine clinic with her, or goes with them to get vaccinated. But you can't talk to every single person. And so there's a variety of reasons, right? And then there was the variety of, well, if they have a more traditional platform like Novavax, the protein-based vaccine, right? They'd be more confident with that because we've stressed how this is a new and exciting platform. Well, maybe we didn't need to stress that it was a new and exciting platform. When they gave the OPV, did they market it as, oh, look, we have a new exciting platform for polio?
No.
Vaccination?
Nope. Of course not.
Okay, well, maybe we should have taken a lesson in their campaign. But we didn't. And so maybe, we should foster pushing through some of those vaccines that would get over that hesitancy because they'd be more confident, right?
Yeah, it makes sense.
And but so, you have to spend a lot of time coming up and discussing and calmly sitting down. And I don't know that that's the scientist's job. I'm not that good at it.
I don't do calm very well either, Amy.
I would not know if it said I spend no time with you whatsoever. And I wouldn't be the target of any of this. So I have no idea.
We have 2 listener picks. One is from Philip, who sends an article on the history of Clemson blue cheese.
What? Okay.
Apparently, Clemson University makes its own blue cheese.
That's cool.
It's a gourmet cheese. They make 25,000 pounds a year. And
Is
it was
that
first,
a lot or a little?
I don't think that's a lot of cheese in terms of world consumption. Anyway, it was first made in 1941, and this gives a history of it. So as Philip says, practical use of microbes. I love blue cheese. Delicious. And we also have one from Jack. Dear TWiV team, I have 2 listener picks. The first one is a blog you probably know about already since it is hosted by Science, but some of your listeners might not know about it. Until last August, it was hosted by Science Translational Medicine. It's Derek Lowe's blog, In the Pipeline. So I used to know this blog before it was hosted by anybody, and Lowe just published it on his own. And Science Translational Medicine decided to publish it. Why hasn't anyone published my virology blog, which I've been publishing for since 2006?
Because you haven't written anything for like 8.5 weeks.
No, I wrote something 2 weeks ago.
Yeah, and where's this week's blog?
Well, I had a guest post this week. I've been writing for how many years? A long time. It's a really good blog. Science, you should have picked me up. I'm not saying Derek isn't good. He's good also. Anyway, to continue, to quote from the blog, Derek Lowe, an Arkansan by birth, got his BA. Why are you shaking your head?
Nothing.
I can see you on video,
Really? That's what this is. Really, nothing gets past. Mine like a steel trap. Nothing gets past you.
No, things get past me. It's fine. He got his BA from Hendrix College, his PhD in organic chemistry from Duke. Before spending time in Germany on a Humboldt fellowship on his postdoc, he's worked for several major pharmaceutical companies since 1989 on drug discovery projects against schizophrenia, Alzheimer's, diabetes, osteoporosis, and other diseases. His experience makes him an expert on the problems of drug discovery, and his reading allows him to discuss a wide range of biomedical subjects with intelligence. His writing is clear, down-to-earth, blunt, sometimes amusing. During the height of the pandemic, his subjects paralleled those on TWiV, such as vaccines and drugs for SARS-CoV-2. Now, you and he are drifting apart as you discuss other viruses and Lowe discusses other diseases. Still, I think there would be a lot of interest to you and your listeners in his blog, and he gives a link. So I'm aware of his blog. I like his writing. I think he does a good job. But I think my blog is good, too.
Well, I didn't know it was a mutually exclusive thing. If you have one blog, you can't have the other.
You can't have too many blogs, Amy.
Yeah,
that's fine.
I'm
just complaining. I'm complaining in a good-natured way.
I understand you're complaining.
There is an index archive of posts, a comment section with responses, blah, 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 sometimes with links. Derek Lowe would be a good guest if you want to discuss new therapeutic agents. All right, this is good, Amy. The second pick is from 1955, the discussion of the new Salk polio vaccine on television by the first secretary of the new Department of Health, Education, and Welfare, Ovida Culp-Hobby, and the Surgeon General of the U.S. Public Health Service, Dr. Leonard Scheel. And there's a video link. I think this discussion is a model of how to convey medical information to the public. Dr. Scheel showed respect for his audience. He described the symptoms of the disease and gave figures for the incidence of paralytic polio, permanent crippling, and death. He described how the vaccine was made, explained why it contained some infectious virus despite inactivation by formaldehyde. He acknowledged that some vaccinated children will develop paralytic polio, and he said that there wasn't enough vaccine to immunize all children in the first summer. This must have been tough for people to hear amid all the celebration. On the day of the press conference announcing the results of the vaccine field trial, April 12th, 1955, people huddled round radios as if listening to the World Series. When the news came, reporters hollered, "It works, it works." Tearful families embraced. Church bells rang out across the nation, and work stopped in offices and factories as celebrations erupted. So there is a way to convey medical information to the public. As Amy just said, it should be done calmly. And our listener also said, and you should have a neutral ground. I totally agree.
Yeah, but one also has to remember the history of polio. I mean, we wrote it recently, right? That there was tremendous fear. You would lock your kid into the house and you would say, "You want to go swimming? Swimming so you can get paralysis? You want to go swimming?"
Yes.
You know, parents were out of their minds with fear.
That's a bit different. But times have also changed. You know, the incident he talks about, there being infectious polio in the vaccine and some kids getting polio, the Cutter incident,
Yes.
that really changed the public view towards vaccine. And that's part of the reason why we're in the situation we are in today. I mean, It didn't start there, but it really amplified the anti-vax sentiment, I think.
Well, yes, it amplified the anti-vax sentiment, but then it kind of went away with Sabin, right?
Yeah, but then it came back because there was Sabin vaccine-associated polio, right?
Yeah, but that took a lot of years to tease out, right?
Yeah, it did.
And even still, if you talk to people today about Sabin, they'll tell you that most people that you talk about that aren't polio people, that are just common people who walk around in the world, they'll tell you that that the polio vaccine is great. There's no side effects. It protects against infection. Right?
Yes, indeed.
And, 1 in a million, you don't usually see. It's not the same. Like my mom can name off of 5 people that she grew up with that had polio.
Yeah, of course.
So, and a childhood disease also has a different scare factor than old people disease.
Yeah, and as you know, COVID is less frequent in children, right?
Well, severe COVID is less frequent in children.
Right? Severe COVID, yeah.
I think that's where the problem began, right? Is that we focused and people made misstatements about, children don't get infected with SARS-CoV-2 because they didn't present a significant number of the early cases of severe disease were not presented in children. I wonder how different the story would have been if the severe cases at the beginning of the pandemic were all under the age of 12.
Oh, totally different. Yes. Good point.
Everybody would be vaccinating, right?
Yeah.
But, when you market it as it's an old person disease over 65, okay, so you lose your grandparent, but you lose your grandparent. You don't lose your child.
All right. On that note, we will wrap up this TWiV number 884. You can find the show notes at microbe.tv/TWiV. You can send us a question, comment, pick of the week, TWiV at microbe.tv. If you enjoy what we do, please support us. We don't charge anyone for our work. We rely on your contributions. Microbe.tv/contribute. We are a nonprofit entity, so your contributions are U.S. federal tax deductible. Amy Rosenfeld is at enterovirus.net, Columbia University. Thank you, Amy.
Thank you.
I always enjoy talking about viruses, and I know you do as well.
Yes, I have nothing else to talk about. I spend my entire day talking about
Well, that's not
viruses.
true. You can talk about bagels and shoes and food and many things, but this is a virus show. I'm Vincent Racaniello.
The first 15 minutes was bagel spreads.
Well, I like to learn about bagel spreads.
Don't put bacon and hot sauce in your bagel.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.